Hello, BNG312 class. This is the biotransport group exercise number 2 pre-lecture here. We did one earlier in the semester about mass balances. This one is going to be about cellular biotechnology, and we're going to talk a little bit about what we've learned, sum things up regarding some of the biotechnology systems that we've learned: animal cells, plant cells, CHO cells, bacteria, yeast, and the like. So there's a whole lot that goes into biotechnology here, and we have touched upon animal cells, plant cells, and fermentations using bacteria or yeast, and there's a lot more than that. And there are a variety of industries that are important in this: pharmaceuticals, med lab, biofuels, food, regulatory, etc. So these all encompass biotechnology, and kind of one of the parts and parcels here of biotechnology is finding a organism that you can use for producing something, whether it's an antibody, whether it's a hormone, whether it's a biofuel molecule, whether it's some other kind of molecule, but something that you can make reliably using some kind of cell. And so that's what the group exercise is going to focus on. So, for example, I'll just read this lot of text here. So the group exercise is essentially this: you have isolated a human tissue cell line that will overproduce a hormone that's needed in the treatment of disease. So under typical cultivation conditions, with some good media, the cells produce 0.1 milligrams of peptide per 10 mils of culture per day. So what's underlined here is what's called the productivity: 0.1 mgs of peptide per 10 mils of culture per day. So a weight, a mass per volume per unit of time. That's your typical productivity. And so that is essentially you've got a primary or a secondary cell line, probably a secondary cell line. This productivity here is what that cell line is making without any kind of alterations. So you wonder if perhaps you could start a company that sells aliquots of this hormone. So you would like to attempt to produce and purify hormone at a pilot scale. But first you want to increase the productivity to 0.5 milligrams of peptide per 10 mils of culture per day. So it's basically a 5-fold increase. And in the future, you envision producing the hormone on a large scale with a productivity of 1 to 2 milligrams of peptide per mil of culture per day. So you're going from a half a milligram per 10 milliliters per day to 1 to 2 milligrams per milliliter per day. So that's quite a jump. And you will also consider expressing the hormone gene in bacteria, which is something you may have to do. But anyway, that's the premise. And so essentially you'll have questions to answer regarding this. And what this is not testing your knowledge per se. This is testing your ability to brainstorm. This is all about brainstorming. So in your groups, don't be afraid to share your ideas, and don't be afraid to write down your ideas. Because those ideas, they may seem crazy at first, but then again, everyone thought the doctor who discovered the Helicobacter pylori that causes ulcers was crazy until he was proven right. So don't be afraid to share your ideas is the common thread on that. And use the concepts you've learned already in this class, especially the last 3 or so lectures, as well as concepts you may have learned in other places to try to come up with answers for the questions. And you are allowed to share your answers with your other group members, write other group members' ideas down as well as your own. In fact, the more the merrier, really, on this. This is literally a lesson in brainstorming, and to see how well we can do it. Okay, and some assumptions to make clear for the assignment. Everything can be improved, so that includes these productivities here. You can say that we can perform genetics, So if one of your ideas is to genetically modify, then that is a valid idea, because we'll assume that it can be done. And the only constraint to scaling up is the productivity. You've got to reach the productivity, which those productivities are underlined in a few slides ago. And thus, you can consider a potentially expensive idea. So if you've come up with some expensive idea that might work, well, that's good in terms of being in the brainstorming stage. Okay, so you will have certain questions to answer. And so since you were the one who isolated it, one of the things you're going to answer is, what do you already know about the cell line? And what could you do to increase the productivity fivefold? And then what could you do to increase the productivity yet even further? Potential pitfalls that could arise in scale up. So as you increase your productivities, what are these potential pitfalls? And how would you transfer the ability to make peptide hormone into, say, E. coli or another bacterium? And again, what would be the pitfalls in doing that? So consider how you could get this done, and consider, well, what bad things could happen if we did do this, in all cases. And you'll see that that's how that is set up on the assignment sheet, on the group assignment sheet that you'll have. Okay, so what you may do, you may obviously consult with each other. That's the point of group exercises. You can also, if you have a laptop or one of your group has a laptop, you can consult the internet. I wouldn't spend too much time doing this. You can kind of clarify some ideas using online tools and such, literature searches, things like that. But again, don't spend too much time. Just focus more on the brainstorming aspect, not on the getting every single minute detail exactly correct, because there is usually more than one correct answer for these things. You can also ask me for clarification. You can bounce an idea off me and say, does that make sense? And I'd be glad to give you my opinion on that. Okay, so the beginning of class, you'll get into your assigned groups, and then I'll hand you the exercise sheet. So then as soon as your group has the exercise sheet, you can get started. You can start brainstorming and start writing down ideas. At the end of the class, we will share one or two ideas about the answers to each question. And I'll try to get representatives from each group to do that. So I'll spend maybe 5 or 10 minutes at the end of class doing that. And then at the very end of class, you'll hand in your work. You'll make sure your names are on it, your name as well as the names of your teammates. Make sure I know which assignment is yours. And the exercise will be graded like a homework. But what this is it's effort-based. So I will pay attention to how well you all worked in groups. I will pay attention to the sort of ideas you put down, and I will go from there and look at what you've written, and you'll be graded based on how hard you worked on it in class. So it's not that we're necessarily looking for the right answer, it's just that I'm looking for, well, how many different ideas and how many valid ideas did you come up with? Okay, so that is essentially what is going on with that. There'll be a group numbering sheet, as always, posted along with this lecture, along with the slides to this lecture, and along with the exercise sheet. Please look at that, and if you have questions about it before the class, feel free to email me. If you have questions during the class, of course, I'll be milling around as usual, ready to answer those questions. And I will see you on Friday.